We're going to look at humoral immunity next. The specific immune response is very specific to which pathogen it's going to react to. So we have our T lymphocytes and B lymphocytes, and they're all going to be very specific for which pathogen they recognize. So in the specific immune response, we've got two branches. We have humoral immunity and cell-mediated immunity. Humoral immunity involves the B lymphocytes, and when the B lymphocyte finds its match, it will start dividing and form plasma cells, which are going to secrete a lot of antibodies. And the antibodies are big proteins that will bind to any pathogens that are out in the body fluids. And the antibodies act like big red flags, and that signals the other white blood cells to come over and get rid of whatever's been tagged. The reason they call it humoral immunity is the old term for body fluids was humors, so that's where that name came from. Cell-mediated immunity is going to involve cytotoxic T lymphocytes, and they're going to find any infected cells, cancer cells, or transplanted cells that they recognize, and then they will destroy those cells. So here's just the picture of the specific immune responses, and we can divide that into two branches. We've got humoral immunity involving our B lymphocytes, which make antibodies, which will bind to pathogens that are out in the body fluid. So here's like a virus. And cell-mediated immunity is going to involve the cytotoxic T lymphocytes, and they will go around and check for their match. So maybe this cytotoxic T lymphocyte recognizes cells that are infected with the measles virus. So if it finds a measles infected cell, it will then kill that infected body cell. And other cytotoxic T lymphocytes will recognize other pathogens or certain cancerous cells or transplanted cells. The word antigen is important to know. An antigen is just anything that the immune system responds to and produces antibodies against. The name comes from combining antibody generator, so that's where antigen came from. An antigen could be a lot of different things. It might be part of a cell wall on a bacterial cell, or maybe it could be the flagellar protein on a bacterial cell, or maybe it's a part of the virus. That could be an antigen. So there's lots of different antigens that are out there. So just some features of the specific immune response that B and T lymphocytes have. So all B and T lymphocytes are specific. They're very diverse. They can form memory cells, and they also have self-tolerance where they ignore the body's own antigens. So let's just take a little bit closer look at these. Each B and T lymphocyte are very specific as far as which antigen they recognize. So you have millions and millions of different B and T lymphocytes. If the B cell or T cell does not recognize that particular antigen, it won't react to it. So it has to find its match before it reacts. How the B and T lymphocyte recognize their antigen is by the use of receptors. The B lymphocytes actually use one of the types of antibodies as their receptor, and they're very specific shape, and they're only going to be recognizing one particular antigen. And then here's the T cells. They each have their own unique T cell receptors, which is not an antibody. The antibodies that are produced by the B lymphocytes are very specific to what they bind to. On the tips of the Y here, this is called the variable region, and this is where it binds to the antigen. And on an antigen, there can be different epitopes. An epitope is just where the antibody binds to. So on this particular antigen, maybe it's E. coli bacteria. Maybe this particular B lymphocyte that produces this antibody recognizes this part of the cell wall of the E. coli bacteria. And then you could have another B lymphocyte that also recognizes E. coli, but it recognizes a different epitope a different part on that particular antigen, the E. coli. We have millions and millions of different B and T lymphocytes, and each one has a unique receptor and will recognize only one particular antigen. And this is just due to genetics. It's very random as far as which shape of B or T lymphocyte receptor is formed, because we never know what we're going to be running across during our lifetime, so we need to be ready for lots of different things. The other thing that B and T lymphocytes have is they're able to form memory cells. So they can remember that they've seen their particular match before. The first time that they're exposed to their match, the B or T lymphocyte will be stimulated to divide, and it's going to produce two different types of cells. The effector cells are going to go and do battle with the pathogen, so they'll fight off the invader. The effector cells are fairly short-lived though. But the memory cells are the other cells that are produced. They will sit on the sidelines and root for the home team, but they're not going to join in the battle. They're long-lived cells usually. Some of the memory cells will last the rest of your life. And those memory cells, if you're exposed to the same pathogen a second time, they will respond very quickly and much more strongly the second time around, and you don't get sick the second time. So let's say that our B lymphocyte in the middle here recognizes the chickenpox virus. So it will be stimulated to divide and form the effector cells, the plasma cells, which will actually make the antibodies to fight off the virus. But these plasma cells are short-lived and won't last a long time. And then the other cells that are produced are these memory B cells, which are long-lived. And for the chickenpox virus, these memory cells will probably live the rest of your life, and so you won't get the chickenpox again. These other B lymphocytes did not recognize the chickenpox virus, so they did not respond. We call this clonal selection. A clone is just a population of cells that are all the same, because when this cell that recognizes the chickenpox virus starts to divide, all of the cells that are produced, the plasma cells and memory B cells, these all recognize the chickenpox virus. So here's the difference between the primary and secondary immune responses. So let's say that you're first exposed to the chickenpox virus, and unfortunately it takes about 10 days for that B lymphocyte that recognizes the chickenpox virus to start dividing, start forming the effector cells, the plasma cells, produce the antibodies, and also produce those memory B cells that'll be there for the next time. So it takes about 10 days before antibodies start getting produced. And in the meantime, during those 10 days, the chickenpox virus has been having a party in your body and causing sores all over the place. So you do get sick on the first exposure. But then you start getting better as you start producing the antibodies and get rid of the virus. And then let's say that maybe years later, you're exposed to the chickenpox virus again. Because you've already have those memory cells that have already been made, they're going to react much more quickly. So notice it only takes them about 3 days. And also they're going to have a much stronger response. They're going to produce a lot more antibodies than you did the first time. So by having this faster and stronger response on the second exposure, you're not going to get sick the second time. And then the fourth thing with the B and T lymphocytes is they have what's called self-tolerance. So we don't want them to recognize any of our body parts and start attacking them. So as the B and T lymphocytes are made, it's just random what type of receptor they make and what they recognize. And some of them actually do recognize body parts. So as they're maturing, the B lymphocytes mature in the bone marrow and the T lymphocytes mature in the thymus. And as the B and T lymphocytes are maturing, they will be presented with different antigens from our body parts. And if any B or T cell recognizes a body part, then they will either be killed by apoptosis, where they commit suicide, or they'll just be turned off. because we don't want to let B and T lymphocytes that recognize body parts out into the body, because then they start attacking us and we end up with an autoimmune disease. All right, let's take a look at humoral immunity. This involves the B lymphocytes. They're going to be producing antibodies that will bind to pathogens that are out in any of the body fluids or the humors. And remember that each B lymphocyte is very specific to which antigen it recognizes. And the B lymphocytes usually are going to need the help of a helper T lymphocyte to activate the B lymphocyte. And when the B lymphocyte gets the green light to start dividing, it's going to produce two types of cells. The plasma cells, which are the effector cells, they're going to be producing the antibodies that will do battle with the pathogen and bind to any of the pathogen it finds out in the body fluids. And then it's also going to produce the memory B cells that will be there for next time, and they're going to react much quicker and stronger the second time so you don't get sick again. Here's just a picture of humoral immunity and how it works. Here's our B lymphocyte. And here's the receptor, and let's say that it recognizes the measles virus. So it says, hey, I found my match, but it doesn't do anything. It just sits there because it needs a double check signal from the helper T cell to make sure it really found its match. So the B lymphocyte will take the measles virus and put it out on its surface on a protein called a class 2 MHC protein, which we're going to talk more about in the next lecture. And this B lymphocyte will act as what's called an antigen presenting cell and present the antigen that it's found, this measles virus, to the helper T cells. So the B lymphocyte will go around to a bunch of helper T cells and see which specific helper T cell also recognizes the measles virus. And when it finds the match, the helper T cell will say, yep, you really did find the measles virus. I recognize it as well. And the helper T cells are going to secrete cytokines, little chemical messengers, and give the green light to the B lymphocyte to start dividing. The B lymphocyte is then going to divide and form two types of cells. The effector cells that are going to go do battle, these are called plasma cells. And they're going to produce lots and lots of antibodies. So they end up churning out about 2,000 antibodies per second, which is a lot. And these plasma cells are not long-lived cells, so they will fight the battle and then usually they'll die off after that. But the antibodies will bind to any of the pathogens that are out in the body fluids. So they're not inside the cells, they're out in the body fluids. And here we've got a virus, very sad, it's been tagged with the antibody, and anything that's bound with an antibody will be marked for destruction and the other white blood cells will come over and get rid of it. And then the other type of cell that's produced are the memory B cells that will not participate in the battle this time, but they're there for the next time you get exposed to the measles virus, and they will react very quickly and strongly so you don't get sick on the second time. And then this is just another picture of what I just drew on that previous picture. And this is just to remind you that the antibodies that are produced by the B lymphocyte that recognizes that measles virus is very specific, and it will only bind to the measles virus antigen. And there's a specific part on the measles virus that it will bind to, and that's called the epitope. There's 5 different classes of antibodies. What makes them different is on the stalk of the Y. This is called the C region or the constant region. So this is what determines the class of the antibodies. So for example, all of the IgG antibodies will have the same C region, whereas the IgM antibodies will have a different C region. And then the variable region, the tips of the Y, that's going to be very specific and only bind to one particular antigen. So make sure you know these 5 classes of antibodies. This is really easy to put on the exam. The IgG antibodies, these are the most common type of antibody found in the blood, and they're the second antibody produced by the plasma cells. The IgM antibodies are actually the first antibodies that are produced by the plasma cells, and then the plasma cells will switch over and start producing IgG. And the reason that is the IgM antibodies, we'll look at a picture in a minute, but the IgM antibodies are huge. They are 5 antibodies stuck together, and so they can bind lots of the pathogen all at once. And then it will switch over and start making IgG antibody a little bit later, because the IgG antibody are just one antibody, and it's much smaller, and so it can kind of get into all the nooks and crannies and finish, clearing out the pathogen. The other thing that's important about the IgG antibody is that it can cross the placenta. So whatever IgG antibodies that mom has will cross the placenta and be given to the baby, and that will actually protect the baby from infections during the first 2 months of life. So that's pretty cool. The IgA antibodies, those are found in mucous membranes. So they're in the mucus, saliva, tears, breast milk. That's where we're going to find the IgA antibodies. The IgA antibodies are very important because they're going to bind to any pathogens that are trying to get in through the mucous membranes, and that'll prevent them from attaching to our cells. And the IgA antibodies are also really important because they're found in the colostrum, mom's first milk that she gives to the baby, and those IgA antibodies will be absorbed by the baby and will also help protect the baby during the first 2 months of life. The IgE antibodies are involved in allergies, and they will stimulate the mast cells and basophils to release histamine, which causes the signs of allergies. And then the IgD antibodies are found on the B lymphocytes. They're the receptors. Here are what the 5 major classes of antibodies look like. So IgG is just one antibody, along with IgD, the receptors on the B lymphocytes are just one antibody. And IgE that are involved in allergies, those are also just one antibody. And you can see the IgM antibodies are humongous. They are 5 antibodies stuck together. And then the IgA antibodies are 2 antibodies that are stuck together. These are the IgA antibodies that are secreted on mucous membranes, and they will bind to pathogens in the mucous membrane and prevent them from binding to our cells and getting inside. And then the other important thing about the IgA antibodies to remember is that they are in the colostrum, that first breast milk, and that will be transferred to the baby and also help protect the baby for the first 2 months of life. And then this is just a picture of the IgE antibodies that are involved in allergies. So people with allergies just have a genetic tendency to produce too many IgE antibodies. And those IgE antibodies get stuck in the surface of the mast cells and basophils. And then whatever you're allergic to, so maybe, I don't know, you're allergic to grass, and when you're exposed to grass again, the grass antigen will then bind to the IgE antibodies on these mast cells and basophils, and that triggers them to release histamine, and then histamine will cause the signs of allergies. Antibodies work in a number of different ways, and we're going to take a look at all of these, how they work. So the first way that antibodies work is through neutralization. So if the pathogen is bound with antibodies, it won't be able to bind to our cells and get inside. So that will block the pathogen from getting inside of us. Agglutination, agglutinate means to clump, and this is when the antibodies bind to a bunch of pathogens. And remember the antibodies, because they've got 2 tips on their Ys, they can actually bind to 2 of the antigens at the same time. And so they end up in this big clump. When we have the antibody bound to its antigen, that's called an immune complex. And this is a big red flag for the other white blood cells to come over and get rid of this. Antibodies can also act as opsonins, so they're going to coat the pathogen and make it easier for the phagocytes to grab onto them and eat them. And remember the other opsonin that we talked about that will coat the pathogens are the complement proteins. And then also we can activate the complement system when the pathogen is coated with the antibodies. That will trigger the first complement protein to be activated, which will trigger the next, and the next. You get that cascade going. And then here the end result is we're making a membrane attack complex, a hole in the bacterial cell, and water will rush in, and then the bacteria will explode. And then also natural killer cells. One of the ways that they recognize an abnormal body cells is that if it's coated with antibodies, they know that that cell is either infected or cancerous and they need to get rid of it. So they'll secrete those porphyrins, make a hole, pour the granzymes in, and tell the cell to do apoptosis.